Plants do things differently. Box 8 to 4 Thinking about the growth of wood. Although the growth of trees and their wood is perhaps the most complex type of growth you will ever try to understand, it really is not too difficult. Use your imagination to picture a complicated object in 3 dimensions and then imagine watching it grow. To start, imagine a tree without any leaves, flowers, or fruits. Now imagine removing the bark from all the woody areas, and then paint all the wood, every bit of the trunk, branches, and roots, but not the twigs or root tips where there is epidermis. If you imagine this correctly, you have one single, continuous coat of paint that indicates where the vascular cambium is, and you have thousands of twig tips and root tips, each with an apical meristem. All active meristems of the tree are now visible in your imagination. Hold that image in your mind, and now imagine you are making bread and have mixed yeast cells with water and flour and then kneaded them until the yeast cells are evenly distributed throughout the dough. Each yeast cell absorbs water and nutrients and then enlarges and divides. Each daughter cell then repeats this process. All parts of the dough contain living cells and all are growing at the same rate. This is diffuse growth, and if we were doing this on the space station without gravity, our ball of dough would grow into a perfect sphere, becoming larger in all directions equally. Plants have localized growth, and at the tip of every living twig and root there is a small mass of dividing cells, the shoot or root apical meristem. They have localized growth. If the meristems were to grow like the bread dough, then the stems and roots would end up looking like lollipops. But all the meristem cells next to existing stem or root tissue act differently from the other meristem cells. They divide less and less and finally stop dividing, having become part of the new stem or root rather than continuing to be part of the meristem. If all meristem cells slowed their rate of division and became stem or root cells, the stem or root would stop growing, and they would not have indeterminate growth. It is important that some meristem cells become added to the growing stem or root, while at the same time some of the meristem cells continue to be meristem cells. Fortunately, our own hair, nails, and skin have localized growth just like this. Let's consider the growth of one of our hairs, as an example. Small blood capillaries at the base of the hair follicle bring water and nutrients to the base of the hair shaft, the area where hair cells are still alive and dividing. The hair meristem cells absorb the nutrients, grow, divide, and then do this again. Those dividing cells that are closest to the hair shaft gradually stop dividing and instead differentiate into dead hair cells. As time passes, more hair meristem cells are added to the base of the shaft and our original cells are thus pushed upward. Each of our hairs grows at its base and is pushed upward out of the hair follicle. The outermost end of each hair is the oldest part, and the base of the hair is the newest. Our hairs do not have growth rings like tree would, but the outer end of each hair is old and the inner end is younger. When growth is localized instead of diffuse, the location of the meristem is important. In roots and stems, the meristem is at the apex, the outermost part, so the apical meristem is pushed away from the plant body by the very shoot or root that it is producing. But the dividing cells of our hairs are located at the base of the hair. They are basal meristems and they remain in the same place. It is the hair itself that is forced to move as new cells are added to it. The same is true for our skin and nails. Remember that one of the themes of this text is to think about alternatives and consequences. I am going to describe an alternative way of growing that does not exist. This is a theoretical alternative that has never evolved, as far as we know. We can imagine that some tree might have 2 vascular cambia located back to back and shaped like a hollow cylinder. The inner cambium would be a wood cambium, its cells would divide, and those cells on the inner side closest to the existing wood would differentiate into new wood cells. We could say this wood cambium is an apical meristem because it is at the apex of the wood it is producing and it is being pushed outward as wood accumulates on its inner side. This wood cambium would be just like a shoot apical meristem except that it would be a cylinder instead of a tiny point, and, for a whole tree, it would have the shape of the paint we imagined in the first paragraph. Lying right to the outside of the wood cambium would be our hypothetical phloem cambium. All its cells would grow and divide, and the cells closest to the mature phloem would differentiate to be new phloem cells, and the rest of the phloem cambium cells would continue to be phloem cambium. Like the cells that produce our skin, nails, and hair, the meristem cells cannot be pushed deeper into the body. Instead, the new tissues must be pushed out. So our phloem cambium would be a basal meristem and the new phloem would be pushed outward, just as our hair and nails are pushed outward. In this hypothetical, non-existent model, our tree would grow wider because of its apical wood cambium and its basal phloem cambium lying back to back. A minor problem is that as new cells are added to the wood becomes thicker and consequently so does its circumference, its surface area increases. Because the wood cambium and the phloem cambium are on the surface of the wood, those two cambia also must become larger. We have a familiar example for understanding this. We have to be extremely careful around a newly born baby because the 5 bony plates that make up its skull are not yet solid. They are still growing at the edges where each piece of bone meets its neighbors. As the baby's brain grows, so does its skull, as the sutures between each plate add new cells to the edges of those plates. The bones in a baby's skull become larger as a meristem of dividing cells along the edges of each plate adds new cells to each plate. And as each plate becomes larger, its perimeter becomes larger, so that meristem along the edges must increase, must become longer. It is just the same for the meristems that cover the surface of a growing cylinder. The baby's skull adds enough new meristem cells so that all parts of the edge of each plate is completely covered in dividing cells and thus each plate grows everywhere along its edge. Now let's go back to the tree. Instead of having 2 meristems back to back, there is just one single layer of cells, just one meristem, the vascular cambium. As its cells divide, sometimes cells on the inner side stop dividing and differentiate as wood, and sometimes instead it is cells on the outer side that stop dividing and become phloem. In both situations it is critically important that some cells continue to divide and act as meristem cells. Because the addition of new cells to the wood causes the wood surface to increase in circumference and because the cambium is pressed tightly against the wood, the cambium's circumference must also increase. The cambium has 2 options. Its cells could occasionally divide to make both daughter cells lie side by side within the cambium and neither one of those would turn into either wood or phloem. In this case the cambium itself would grow and have more cells. By doing this often enough the cambium will always have enough cells to completely cover the wood cylinder, no matter how broad it becomes and no matter how huge its circumference. Alternatively, rather than dividing to make new cambium cells, the cambium cells could just enlarge and become bigger cells. But as the tree continues to grow the cambial cells will also need to grow, and at some point they would reach a size that is just too large for a living, dividing cell to survive. At that point cell division would have to stop, and then the tree could no longer increase in width. This type of cambium did indeed evolve in one group of plants, and the trunks it produced never grew to be very wide. To summarize, shoots and roots of woody plants become longer as new cells are added to their apices in a form of localized, apical growth. Woody shoots and roots become wider through the action of a single vascular cambium that is only one layer of cells thick, and that layer produces xylem to its inner side and phloem to its outer side, and it also produces new cambial cells. Each year, as the trunk or branch or root becomes longer, a new section of vascular cambium must differentiate, and it attaches itself to the pre-existing cambium, and in the following year this will be repeated. At any given time the tree has only one cambium. When trying to understand how trees grow, it is important not to make too much comparison with our bones. We refer to wood as a tree skeleton or that wood supports a tree the way our bones support and strengthen our body. But keep in mind that our bones are composed of living cells, most of which are encased in a layer of calcium phosphate. This may sound like a wood cell with a lignified secondary wall, but bone cells are alive and can remodel themselves, and bones are constantly taking some cells from certain spots, adding other cells to other spots, and remodeling the calcium. Broken bones heal if set properly because our bodies can readjust the cells and the calcium phosphate, but the wood in a broken branch never heals. Some new wood may be laid down around it if the cambium survives, but the broken splinters inside never heal. And do not compare our growth in height with that of a tree. As we grow taller, the ends of our bones are constantly being reshaped and remodeled. New cells are added to the ends, and old cells are removed from other areas. This never, ever happens in plants. New wood cells are never added to the top end or bottom end of an existing piece of wood. Instead, a piece of wood only grows wider through its vascular cambium. A branch or trunk becomes longer only because an apical meristem makes a new section of twig, and at some point that develops a new section of cambium that then starts to make wood, and this new cambium is attached to the end of the old cambium. At the next level. 1. Wood ecology. Wood of many different types has evolved by alterations in the diameter and number of vessels, thickness and lignification of fiber walls, presence or absence of tracheids, and modifications of perforation plates. Each of these factors affects the way a plant is adapted to a particular ecology. I recommend the book Ecological Strategies of Xylem Evolution by S. Carlquist. 2. The beauty of wood. Many types of wood are used by artists to create beautiful objects such as bowls, trays, boxes, and furniture. An internet search will produce many interesting articles. I recommend the book Identifying Wood, Accurate Results with Simple Tools by R. Bruce Hoadley as an excellent introduction into the ways a person identifies unknown samples of wood. It also describes many types of artisanal wood. A second recommendation is Atlas of Woody Plant Stems, Evolution, Structure, and Environmental Modifications by F. H. Schweingruber, A. Borner, and E. D. Schulz, which has hundreds of magnificent photographs and light micrographs of wood structure and the plants that produce the wood. It is superb. Almost any arts and crafts fair has people selling items constructed from beautiful woods. Give some of these a close look and see if you can identify the woods. If not, ask the craftsman. 3. Wood as a source of lumber and chemicals. Wood is extremely useful to us as a source of lumber, paper, and chemicals such as resin and turpentine. Wood is essential to the sound of musical instruments such as pianos and violins. Wood can be sawn, chipped, ground to sawdust, glued, bent, and treated in many other ways. Some of these processes are modern, others are ancient. All will give you a deeper understanding of wood. In almost all cases, the anatomy of a piece of wood determines whether it will be suitable or useless for a particular product. Summary. 1. Cells and tissues produced by apical meristems are primary tissues, whereas those produced by the vascular cambium and cork cambium are secondary tissues. 2. The vascular cambium contains fusiform initials that produce the elongate cells of secondary xylem and phloem, and ray initials that produce ray cells. 3. Secondary xylem contains the same types of cells as primary xylem, but the arrangement differs. Growth rings with early wood and late wood are usually present. 4. In an old trunk, branch, or root, the central xylem is heartwood, it is dry and its parenchyma has died. The outer, younger xylem is sapwood, it is involved in water conduction and its parenchyma is alive. 5. Cork cambium produces resistant cork cells, the cork and all exterior tissues are the protective outer bark. Lenticels permit oxygen diffusion into the organ. 6. Palms appear woody because their numerous vascular bundles contain many fibers. Certain other monocots have anomalous secondary growth, in which a vascular cambium produces secondary vascular bundles. Important terms. Phellem self-elogen primary tissues radial system ray initials rays ray tracheids reaction wood ring porous sapwood secondary phloem secondary tissues secondary vascular bundles secondary xylem softwoods storied cambium tylosis. Vascular cambium wood. Review questions. 1. Growth by apical meristems has been previously described. In woody species, additional tissues are produced in the stem and root by two other meristems, the and the. The new tissues themselves are and. They are the secondary tissues, and they constitute the plant's secondary body. 2. Name at least 3 examples of woody plants that are trees and 3 that are shrubs. 3. Many herbs live for several years. How do they respond to the bottleneck of the limited conducting capacity of the first-year stem? 4. Which groups of plants have secondary growth? Which never do? 5. Imagine a tree that has a radius of 20 centimeters and that produces a new layer of wood 0.5 centimeters thick. What is the cross-sectional area of the new layer of wood? If each leaf needs 0.1 centimeters of wood to supply it with water, how many leaves can the tree have? If the tree produces a new ring of wood next year that is again 0.5 centimeters thick, how many leaves can the tree have next year? 6. Woody plants are almost always perennial plants, often living for many years. Describe some advantages of this with regard to a plant's ability to occupy a favorable site. Describe some disadvantages with regard to how long a plant must survive pathogens and harsh conditions. 7. It must be difficult for secondary growth to arise by evolution. How many times has it evolved in the 420 million years that vascular plants have been in existence? How many of those have become extinct? 8. What is the name of the vascular cambium that arises within vascular bundles? Between vascular bundles? After the two have formed, what is the shape of the vascular cambium? Is it a series of strips that are aligned up and down the trunk or root, or is it a complete cylinder? 9. Imagine a tree that is growing in springtime. All woody parts that were present last year have a vascular cambium, but all of the twigs that are brand new have just primary growth with vascular bundles. When will a vascular cambium form in these twigs? Will it connect to the old, pre-existing vascular cambium, or will it remain separate? Describe how this occurs. 10. Figure 8.3 is complex but easy to understand. Figure 8.3b shows a tree trunk, and this year is its 4th year of growth. A transverse section at the base of figure 8.3b would show how many layers of wood? Would a transverse section through the top of figure 8.3b show any layer of wood this year? Would it show a layer of wood at the end of next year? Would there be any transverse section that does not show the pith and primary xylem, that is? Are pith and primary xylem ever lost or destroyed by the formation of wood? The orange layer is labeled provascular tissue in the top right of figure 8.3b, but what is it labeled in the middle and lower right diagrams? 11. What are the 2 types of cells in the vascular cambium? Can either type be converted into the other? 12. Look at figure 8.6. Part shows the lower half of a fusiform initial, and part shows the same cell after it has divided and one of the daughter cells is developing into a xylem cell. Did the fusiform initial divide with a paraclinal wall or an anticlinal wall as it went from part A to part B? Part shows that the same fusiform initial has now produced a second cell that is developing into a phloem cell. Was that phloem cell produced by a paraclinal or an anticlinal division of the fusiform initial? Part shows the fusiform initial dividing and forming a second fusiform initial. Is this division occurring by a paraclinal wall or an anticlinal wall? 13. Are fusiform initials parenchyma cells, collenchyma cells, or sclerenchyma cells? Do they have chloroplasts, chromoplasts, or proplastids? 14. Look at figure 8.8. Vascular cambium is not present. It was located far above the top of the micrograph, but you can see there are 6 rows of wood cells. How many fusiform initials were involved in producing the wood visible in this figure? Look at all the cells in any particular row. Were all the cells of that row made by divisions of one fusiform initial or by many different fusiform initials? 15. Are ray initials longer or shorter than fusiform initials? Can they undergo both periclinal and anticlinal divisions or just one or the other? 16. Ray initials are typically grouped together in short vertical rows only one cell wide. 2 cells wide or many cells wide. 17. True or false, typically a vascular cambium never has large regions of only fusiform initials or ray initials. 18. What fraction of cells formed to the interior of the vascular cambium develop into secondary xylem, known as wood, less than half, exactly half, or more than half all cells? 19. In gymnosperms, what types of cells occur in the axial system? Which cells are very rare? Wood of conifers is called because a certain type of cell is absent from their axial system. Which type of cell is that? 20. In basal angiosperms and eudicots, what types of cells occur in the axial system? What is the only type of cell present in the rays of eudicots? The wood of eudicots is called because it has the type of cell lacking in conifers. Which type of cell is that? 21. What types of cells are derived from fusiform initials? What types of cells are derived from ray initials? Is it theoretically possible to have a vascular cambium without ray initials or without fusiform initials? 22. Look at the woods of figure 8.13 in part. All vessels have about the same diameter and are narrow, but the wood in part has some very wide vessels and some narrow ones. The answer is not in the text, but would you think that these two species have different water conducting needs? Is it possible that one species lives in areas with very rainy spring times and the other lives in an area that is rather dry all the time? 23. What is a growth ring? How does early wood differ from late wood? Do all species of wood show strong differences between these two phases of a growth ring? 24. Why do some people prefer the term growth ring rather than annual ring? What can sometimes happen if a summer is unusually cool? Do you think this occurs more frequently in California or in Alaska? 25. If especially wide vessels are produced early in the growing season and only narrow, sparse vessels are produced later, the wood is said to be porous. However, if vessels have similar size and abundance throughout a growth ring, the wood is porous. Name 3 examples of each type. 26. What changes occur as sapwood is converted to heartwood? 27. What is a tylosis? How does formation of tyloses slow the spread of fungi in wood? Why doesn't a tree make tyloses in vessels that are still conducting water? 28. The thickness of sapwood is some indication of how long a growth ring of wood is able to function. Look at Table 8.3 about how long does wood of catalpa function? How about wood of wild black cherry or honey locust? 29. Because secondary phloem is formed from the vascular cambium just as secondary xylem is, it too has an system and a system. Why do the size, shape, and number of phloem rays match those of xylem rays? 30. In a cross-section of a tree, where are the oldest growth rings, in the outer region or nearer the pith? Where is the oldest secondary phloem, near the outside of the tree or near the cambium? 31. What causes the outermost tissues of a woody stem or root to become pushed outward and expanded? 32. The layer of cells that produces cork has a technical name and an ordinary name. Give each name. 33. Periderm consists of at least 2 types of cells. Sometimes a third is present. What are these 3 types of cell? 34. Why does a layer of periderm offer only temporary protection? When the plant makes a new layer of cork cambium, does it make the new layer to the outer side of the failing periderm or to the inner side of it, deeper in the secondary phloem? 35. In which tissues does the first cork cambium form? When does it usually arise? In which tissues do later cork cambia form? 36. Are geraniums herbs or woody plants? Do they ever form bark? 37. What type of plant produces the cork used for wine bottles and cork boards? Pines, oaks, maples, eucalyptus, or none of these? 38. What is the function of lenticels? How are intercellular spaces important for this function? 39. Do roots form wood and bark, or are these secondary tissues present only in stems? 40. Describe the anomalous secondary growth in roots of sweet potatoes. What is the selective advantage of this unusual type of secondary growth? 41. Describe the formation of included phloem. How did the included phloem of Eyersign in Figure 8:30a become surrounded by xylem? 42. Describe the secondary growth of monocots like Joshua trees and dragon trees. What are secondary vascular bundles? 43. Describe the growth of a palm seedling. For the first few years of life, the seedling becomes wider, but without secondary growth, how is it able to do this? Each adventitious root adds something. What is it? If the palm stem has 100 vascular bundles and then forms 5 new adventitious roots with 8 bundles each, how many vascular bundles can the stem have above these new adventitious roots? Would the stem be wider or narrower above these adventitious roots? 44. Why can a monocot like an iris branch and increase its number of leaves? Is the fact that the shoot is a rhizome with adventitious roots important? Is water transported from one end of the shoot to the other? 45. What is the name of the analysis of tree rings? How is that used to study past climates? How is it used to establish the date when ancient buildings and ships were constructed? 46. In tree ring analysis, what is a floating sequence? Why has the sequence for the Middle East been floating? Whereas we know the exact dates of each ring in the sequences for North America and Europe. What is so important about the volcanic eruption in 1628 BCE? Do you think it is possible that the unusual ring in North American tree sequences was produced by a volcanic eruption in North America and the unusual ring in the Middle East sequence was caused by a different phenomenon that occurred in a different year? If so, then has the floating sequence been anchored? Design credits: Hummingbird, Tongo 58 Moment Getty, Green Plant Cells, Shutterstock Incorporated Natalia Ora, Purple Tulip, Shutterstock Incorporated Marie C. Fields, Dandelion, Shutterstock Incorporated Daniel Kreisel, Poppy, Shutterstock Incorporated Saryuri, Plant Icon, Shutterstock Incorporated Vector, Digging Man Icon, Shutterstock Incorporated ZArt. Chapter 9. Flowers and Reproduction. Chapter Opener Image. These are the dry fruits of a small wild mustard. In mustard, each fruit has a thin septum and seeds are attached along the edges. Here, the walls of the fruits have fallen away, freeing the seeds, but several have not yet been blown away. With their disc-like shape, the seeds probably float on water, and rain will thus spread them away from the parent plant. Notice several tiny, brown seed-like structures. These started to develop as seeds but failed for some reason. Outline. Concepts. Asexual reproduction. Sexual reproduction. The plant life cycle. Flower structure. Gametophytes. Fertilization. Embryo and seed development. Fruit development. Flower structure and cross-pollination. Cross-pollination. Stamen and style maturation times. Stigma and pollen incompatibility. Monoecious and dioecious species. Animal pollinated flowers. Wind pollinated flowers. Oviposition. Inflorescences and pollination. Fruit types and seed dispersal. True fruits and accessory fruits. Classification of fruit types. Box 9 to 1 Plants and people, flowers, fruits, seeds, and civilization. Learning objectives. After reading this chapter, students will be able to discuss the advantages and disadvantages of asexual plant reproduction. Discuss the advantages and disadvantages of sexual plant reproduction. Explain one method of asexual reproduction. Identify the structures involved in the plant's sexual reproduction cycle. List the 4 types of floral appendages. Describe microgametophytes and megagametophytes. Discuss endosperm development. Name the stages of embryo development. Compare cross-pollination, self-pollination, wind pollination, and animal pollination. Explain the advantages and disadvantages of inflorescences. Describe ways to classify fruits. Did you know? Flowers contain the organs and tissues angiosperms need for sexual reproduction. Cones are equivalent structures in conifers. In addition to sexual reproduction, many plants also reproduce asexually, producing progeny that are clones of themselves. Seeds are always produced within a fruit or a cone, there are no exceptions. Pollen grains are complete plants, they have less than a dozen cells, have no leaves, no roots, and no stems, but are in fact complete plants. Concepts. Reproduction can serve 2 very different functions, producing offspring that have identical copies of the parental genes or generating new individuals that are genetically different from the parents. Under certain environmental conditions, species that are genetically diverse survive better than genetically homogeneous species. Under other conditions, just the opposite is true. A plant that has been able to survive and grow to reproductive maturity is relatively well adapted to its location, so any progeny that are genetically identical to it are at least as well adapted as it is. Any progeny that are not genetically identical to the parent may or may not be well adapted to the conditions to which the parent is adapted. If the environment is stable during several lifetimes, it is selectively advantageous for an organism to reproduce asexually by budding or sending out runners, thus producing new similarly adapted individuals. If the environment is not stable, however, such offspring may find themselves in conditions for which they are poorly adapted. If all are identical, all may die. Instability of the environment can result from many factors. Irregular climatic events, such as unusually severe freezes, droughts, floods, or hurricanes, also disrupt plant communities. If all members of a species are equally susceptible to low temperatures, all may be killed by a rare freeze, but if the species is genetically diverse, some individuals may survive. Even though most die, the few survivors may be sufficient to repopulate the site. A current concern is that we are adding so much carbon dioxide to the atmosphere that our climate is changing too rapidly for many plant and animal species to be able to adapt. With asexual reproduction, progeny are never more fit than the parent, but during sexual reproduction, sex cells of one plant combine with those of one or several others, resulting in many new gene combinations. Sex cells are so small that many can be produced by a single plant, and many new combinations of genes can be tested rather inexpensively. For example, a single large tree can produce thousands of flowers and millions of pollen grains, each genetically unique, yet the tree uses only a few grams of carbohydrate, protein, and minerals. Similarly, thousands of egg cells can be produced using only a small amount of resources. The pollen from one plant can be blown or carried by pollinators to the flowers of hundreds or thousands of other plants, and one plant may receive pollen from numerous other individuals. The thousands of seeds produced by a single sexually reproducing plant represent thousands of natural genetic experiments. During seed and fruit maturation, those embryos with severely mismatched genes abort and use no further resources. The tree finally produces hundreds or thousands of fruits and seeds. the total reproductive effort may be a significant drain on the tree's resources, but it produces numerous embryos, many of which are at least as genetically fit as the tree is and perhaps even more fit. In both stable environments and changing ones, sexual reproduction provides enough diversity of progeny that at least some are well adapted. Table 9. 1. Sexual and asexual reproduction. Figure 9. 1. These apple fruits have developed from flowers, and the seeds inside the fruits developed from egg cells inside the flowers. All egg cells produced by this tree have similar genes, but sperm cells were necessary to fertilize the eggs, and bees may have brought many types of sperm cells from many different apple trees. Although every seed here has the same maternal parent, it is possible that all the seeds have different paternal parents. The seeds are not genetically identical. These anthers of a lily flower are each releasing thousands of pollen grains. Because they were produced by meiosis with crossing over, each pollen grain is at least slightly different genetically from all the others produced by the same plant. As a further example, think of sexual reproduction in humans. The children produced by a particular couple are variable, not identical to each other or to either parent. Some of the children may have a particularly advantageous combination of genes and be more healthy or intelligent or athletic or creative than either parent. Others may have combinations of genes that result in congenital problems such that the children survive only through medical help. Most children are more or less the same as the parents. The diversity is important. Sexual reproduction also has negative aspects. 2 individuals are required, and sex cells must move from one plant to another. In seed plants, pollen may be carried by wind, insects, and birds, but each results in the loss of many pollen grains or the need to produce nectar. Furthermore, potential sex partners may be widely scattered. For example, in a population of trees, those few individuals growing at the highest altitudes may have no neighbors, whereas those growing at lower altitudes have numerous close neighbors. The flowers of the highest individuals may receive no pollen and thus produce no seeds during some years. In contrast, plants that reproduce asexually can do so at any time, even when completely isolated. Some flowering plants are self-fertile and can undergo self-pollination, but they lose the benefit of receiving new genes from another plant. Some plants reproduce both sexually and asexually. Figure 9. 2 strawberries have flowers and sexual reproduction involving genetically diverse embryos and seeds, but they also spread rapidly and asexually by runners. Bamboos are perennial grasses that flower and set seed only occasionally, but their rhizomes grow vigorously and establish many new plants asexually. Kalanchoes produce large numbers of seeds each year, but they also produce such large numbers of plantlets along their leaf margins that they can be weeds in both nature and in greenhouses. Figure 9. 2 Kalanchoe plants are called maternity plants because they produce plantlets complete with stems, leaves, and roots along their leaf margins. Although hundreds of plantlets can be produced, all nuclear divisions are mitotic, duplication division, and thus, all of these plantlets are genetically identical to the parent, none is superior. Chalice, species of Opuntia, have branches only weakly attached to the trunk. If an animal brushes against a branch, the spines stick to the animal and the branch is pulled from the plant. After the animal dislodges the branch, it roots and grows into a new plant. Long-distance distribution almost as extensive as that of seeds can occur. All the leafless trees with white bark are quaking aspen in winter. Almost certainly, they are all parts of the same plant, each having grown from an adventitious shoot bud on a widely spreading root system. Some aspen clones fill entire valleys or hillsides. All grapes used for wine are the same species, but there are hundreds of varieties, each of which must be propagated vegetatively to ensure every plant produces grapes with exactly the same flavor. Here you can see an entire field planted just with cuttings. Every plant is a clone and each is genetically identical to the others. The clonal nature of this dog cholla is easy to see. The oldest part of the plant was located at the center and then its branches began growing outward. The old, central parts have died, but the younger, newer branches on the edge continue to grow outward and the ring becomes larger. Seeds, which are produced by sexual reproduction, often have a means of long-distance dispersal. Strawberries are eaten and the seeds later defecated. Bamboo fruits and seeds are carried by winds. The consequence of this is that seeds may become widely scattered and germinate in numerous diverse sites, each site differing from the others in its microclimate, soil conditions, and exposure to predators and pathogens. Of course, a seed carrying an embryo with a combination of genes selectively advantageous for growth in a dry site may land in a wet site and not survive, but with the large numbers of seeds produced, it is statistically probable that some seeds will land in sites for which they are well suited. In contrast, new plants that are produced asexually are usually not capable of long-distance dispersal. Runners, rhizomes, and plantlets result in new plants that become established in the same microhabitat as their parent. After a single plant becomes established in a suitable site, by reproducing asexually it can quickly fill the area with replicas of itself, all of which are as fit as it is. Asexual reproduction. Within angiosperms, numerous methods of asexual reproduction have evolved. One of the most common is fragmentation. A large spreading or vining plant grows to several meters in length, and individual parts become self-sufficient by establishing adventitious roots. If middle portions of the plant die, the ends become separated and act as individuals. Certain modifications improve the efficiency of fragmentation. In many cacti, branches are poorly attached to the trunk, and the plant breaks apart easily. The parts then form roots and become independent. In some members of the saxifrage, grass, and pineapple families, plantlets are formed where flowers would be expected. These look like small bulbs and are called bulbils. In willows and many thistles, adventitious shoot buds form on roots and then grow into plants. Adventitious buds may grow out even while the parent plant is still alive, and a small cluster of trees may in fact consist of just a single individual. A grove of aspens that covers several acres in Utah has been discovered to be a single plant. Sexual reproduction. Sexual reproduction in angiosperms involves flowers, which produce the necessary cells and structures. To understand flower structure, one must first understand the plant life cycle. The plant life cycle. The life cycle of mammals such as humans is simple. Diploid adults have sex organs that produce haploid sex cells called gametes, either sperms or eggs, by meiosis. Individuals that produce sperms are called males, of course, and individuals that produce eggs are females. One sperm and one egg are brought together, forming a new single diploid cell, the fertilized egg or zygote, which then grows to become a new individual that is diploid and resembles its parents. In plants, the life cycle is more complex. The plants you are familiar with, trees, shrubs, and herbs, are all just one phase of the plant life cycle, called the sporophyte phase or sporophyte generation. A critical factor is that sporophytes are always diploid, like most adult animals, and they have organs with cells capable of undergoing meiosis. In animals, meiosis results in haploid gametes, but in plants, it results in haploid spores. The difference between gametes and spores is great. Gametes can fuse with other gametes in a process called syngamy or fertilization, thereby producing the diploid zygote. A gamete that does not undergo syngamy dies because it cannot live by itself and usually cannot grow into a new, haploid individual. Plant spores are just the opposite. They cannot undergo syngamy, but each undergoes mitosis and grows into an entire new haploid plant called a gametophyte. It is called a gametophyte because it is the plant that produces gametes. During sexual reproduction, when a sporophyte reproduces, it does not produce a new diploid plant like itself, but rather a haploid plant. Furthermore, in all vascular plants, a haploid gametophyte does not even remotely resemble a diploid sporophyte. It is a tiny mass of cells with no roots, stems, leaves, or vascular tissues, but it is an entire plant. Gametes are formed by the haploid plants by mitosis, not meiosis. The gametes then undergo syngamy, forming a zygote that grows into a new, diploid sporophyte, and the life cycle is complete. Mammalian gametes are of two types, small sperm cells that swim and large eggs that do not. This is also true of many plants and is known as oogamy. In oogamous plants, just as in oogamous mammals, sperms are produced by one type of individual and eggs by a different type of individual, hence, there are male or microgametophytes and female or megagametophytes. The two types of gametophytes have grown from two types of spores, microgametophytes from microspores and megagametophytes from megaspores. Having two types of spores is known as heterospory. Typically, just one kind of sporophyte occurs in a life cycle, and it produces both microspores and megaspores. A life cycle like this, with two generations, sporophyte and gametophyte, is said to be an alternation of generations. Because gametophytes do not resemble sporophytes at all, this is an alternation of heteromorphic generations. This is a complex life cycle, with at least 3 distinct plants. The human life cycle, like that of all other animals, does not have anything equivalent to the haploid generation. Figure 9, 3 Most flowers have 4 types of structures: sepals, petals, stamens, and a pistil. The pistil shown here is actually composed of 5 parts called carpels that have merged into one structure. In the anthers of stamens, the central cells undergo meiosis and each produces 4 daughter cells called microspores or pollen grains in the flower's central organs. The carpels or ovules, each containing only one cell that undergoes meiosis. Often 3 of the daughter cells die and the one survivor becomes the megaspore. Flower structure. A flower is basically a stem with leaf-like structures, so almost everything discussed for vegetative shoots also applies to flowers. Flowers never become woody. Secondary growth does not occur in flowers. The flower stalk is a pedicel, and the very end of the axis, where the other flower parts are attached, is the receptacle. There are 4 types of floral appendages: sepals, petals, stamens, and carpels. Most flowers have all 4 types and are complete flowers. They typically have 3, 4, 5, or more appendages of each type. For example, lilies have 3 sepals, 3 petals, 3 stamens, and 3 carpels. It is not uncommon for flowers of certain species to lack 1 or 2 of the 4 basic floral appendages, thus being called incomplete flowers. Flowers of begonia are incomplete, lacking petals but having either stamens or carpels. Flowers of pigweed have all parts except petals. Sepals. Sepals are the lowermost and outermost of the 4 floral appendages. They are modified leaves that surround and enclose the other flower parts as they mature. Sepals are typically the thickest, toughest, and waxiest of the flower parts. They protect the flower bud as it develops, keeping bacterial and fungal spores away, maintaining a high humidity inside the bud, and deterring insect feeding. Sepals also protect the flower from nectar-robbing insects and birds. If flower buds develop in a protected position such as beneath a spiny cover or surrounded by regular leaves and branches, sepals are less important for protection and may be small or absent. It is not uncommon for sepals to be colorful and help attract pollinators. All the sepals together are referred to as the calyx. Figure 9, for all seed plants produce 2 types of gametophytes. Microspores develop into microgametophytes. The microgametophyte body is so small that it has just 3 cells and fits inside the pollen cell wall. In angiosperms, pollen grains convert to microgametophytes before the anther opens and sheds the pollen. The microgametophyte produces 2 sperm cells located within its own protoplasm. The megaspore develops into a megagametophyte. It is slightly larger than a microgametophyte and has 7 cells, one of which has 2 nuclei. One of the cells is the egg. The megaspore and megagametophyte are never released from the flower of the parent sporophyte, so they appear to be just sporophyte tissues, not one plant growing inside another. After one sperm cell fertilizes the egg cell, the new egg cell nucleus is diploid and the cell is a zygote. It develops, by mitosis, into a new sporophyte, shown here as the embryo in an immature seed. The entire ovule develops into a seed, the ovary develops into a fruit. Petals. Above the sepals on the receptacle are petals, which together make up the corolla. Sepals and petals together constitute the flower's perianth. Petals are also leaf-like, being broad, flat, and thin, but they differ from leaves in that they contain pigments other than chlorophyll, have fewer or no fibers, and tend to be thinner and more delicately constructed. Petals are important not merely in attracting pollinators, but rather the correct pollinators. Each plant species has flowers of distinctive size, shape, color, and arrangement of petals, allowing pollinators to recognize specific species. Sexual reproduction results only if pollen is carried to other flowers of the same species. It cannot occur efficiently if pollen is carried to other plants indiscriminately. If a flower has a distinctive pattern and offers a good reward such as nectar or pollen, the pollinator is likely to search for and fly to other flowers with the same pattern, thereby enhancing cross-pollination. Mutations are advantageous selectively if they cause flowers to have characters easily recognized by their pollinators. In addition to visible colors, many flowers have pigments that absorb ultraviolet light, creating patterns only insects can see. Without light, colors cannot be seen, and night-blooming species have white flowers lacking pigments. Their petals produce volatile fragrances, and insects and bats follow the aroma gradient to the flower. Pollen is carried by wind in many species and by water in a few. Typically, petals do not develop in wind-pollinated species. They cannot attract wind, so mutations that inhibit their differentiation prevent plants from wasting resources constructing non-functional structures. Stamens. Above the petals are stamens, known collectively as the androecium. Stamens are frequently referred to as the male part of the flower because they produce pollen, but technically, they are not male because the flower, being part of the sporophyte, produces spores, not gametes. Only gametes and gametophytes have sex. Stamens have two parts, the filament and the anther, where pollen is actually produced. As part of the sporophyte, the anther is composed of diploid cells, and in each anther, long columns of tissue become distinct as some cells enlarge and prepare for meiosis. These microspore mother cells or microsporocytes continue to enlarge and then undergo meiosis, each producing 4 microspores. Neighboring anther cells, in a layer called the tapetum, act as nurse cells, contributing to microspore development and maturation. Microspores initially remain together in a tetrad, but later separate, expand to a characteristic shape, and form an especially resistant wall. They are then called pollen. The anthers open along a line of weakness and release the pollen. The wall of a pollen grain is a cell wall, however, it is quite complex structurally. It has an inner layer called the intine, composed of cellulose, and an outer layer called the exine that consists of the polymer sporopollenin. It has one or several weak spots, germination pores, where the pollen opens after it has been carried to the stigma of another flower. Sporopollenin is remarkably waterproof and resistant to almost all chemicals. It protects the pollen grain and keeps it from drying out as it is being carried by wind or animals. The exine can have ridges, bumps, spines, and numerous other features so characteristic that each species has its own particular pattern. In many cases, it is possible to examine a single pollen grain and know exactly which species of plant produced it. Because sporopollenin is so resistant, pollen grains and their characteristic patterns fossilize well. By examining samples of old soil, botanists can determine exactly which plants grew in an area at a particular time in the ancient past. Figure 9.5 Diagram of the life cycle of an angiosperm. A flowering plant life cycle showing the actual structures involved. Figure 9.6 In most species of flowering plants, the stamens and carpels are produced together in the same plant, but in a few species like this begonia, stamens occur in flowers without carpels, whereas carpels occur in stamenless flowers. In species in which stamenless flowers occur on one plant and carpel-less flowers occur on a separate one, the two types of plants look so much alike that, in contrast to mammals, they usually cannot be identified as stamenate or carpellate without looking at the flowers. In both types of begonia flowers shown here, the pink structures look like petals but are instead modified sepals. True petals are absent. Figure 9.7 The sepals of this rose form a tight covering over the rest of the flower as it develops, protecting the inner parts. When the microspores and megaspores are ready, the sepals bend outward and the flower opens. Sunflowers and daisies are actually clusters of many small flowers that together have the appearance of a single flower. In this case, the entire cluster of flowers is protected by a set of bracts that look and function like sepals. It is not necessary for the individual flowers to have protective sepals. Petals, like these of the mallow, are leaf-like in being thin and flat, but in most species, they are less leaf-like than sepals. Leaves and petals of arbutus can be compared here. These petals are highly modified, and the 5 petals of each flower have fused together, forming a tubular corolla. Figure 9.8 Some flowers do not produce nectar. The pollinator eats part of the stamens and pollen instead. This is more expensive metabolically for the plant because pollen is rich in protein, whereas nectar is composed of carbohydrate. In these flowers of cannonball tree, the lower stamens are modified for edibility. They are large and showy and produce little or no pollen. The pollen-producing stamens are small and inconspicuous, easily overlooked by pollinators. Figure 9.9 Distinctive patterns like this are known as nectar guides. They direct the pollinator to the nectar and position the animal to pick up pollen. Carpels. Carpels constitute the gynoecium, located at the highest level on the receptacle. Carpels have 3 main parts: a stigma that catches pollen grains, a style that elevates the stigma to a useful position, and an ovary where megaspores are produced. A flower can have 0 to many carpels. Usually they are fused together into a single compound structure, frequently called a pistil. Inside the ovary are placentae, regions of tissue that bear small structures called ovules. Ovules have a short stalk, called a funiculus, that carries water and nutrients from the placenta to the ovule by means of a small vascular bundle. The ovule has a central mass of parenchyma called a nucellus. Around the nucellus are 2 thin sheets of cells that cover almost the entire nucellus surface, leaving only a small hole at the top. As in anthers, some nucellus cells, usually only 1 in each ovule, enlarge in preparation for meiosis. These are megaspore mother cells or megasporocytes. After meiosis, usually 3 of the 4 megaspores degenerate, and only 1 survives, becoming very large by absorbing the protoplasm of the other 3. Megaspores differ from microspores because the ovule and the carpel do not dehisce and the megaspore remains enclosed inside the carpel. Figure 9.10 Transverse section through an anther of a lily, showing microsporocytes in the meiotic division that will produce haploid microspores. The surrounding cells constitute the tapetum and aid in the maturation of the pollen grains. After meiosis, the 4 microspores remain together temporarily, but as they develop their specialized pollen cell wall, they usually separate from each other. Table 9.2 Stamen and carpel structure. An ovule develops into a seed after its egg is fertilized, and the surrounding ovary develops into a fruit. Each ovary might have either 1 or many placentae, each bearing 1 or many ovules. Ovaries with just a single ovule develop into fruits with a single seed. Many seeded fruits have numerous placentae, each with many ovules. Orchid pistils always consist of 3 carpels, each of which might have tens of thousands of ovules, and some orchid fruits are estimated to contain up to 1 million tiny seeds. Aside from bearing ovules, the rest of the carpel is at least somewhat leaf-like. Figure 912a shows 3 carpels fused together as a pistil. The outermost layer is basically leaf lower epidermis and typically has stomata. The innermost layer is upper epidermis, and all middle layers are mesophyll and vascular bundles. Gametophytes. Microgametophyte. Microspores develop into microgametophytes. In all angiosperms, each microgametophyte is very small and simple, consisting of at most 3 cells located within the original pollen cell wall. The microspore nucleus migrates to the side of the pollen grain and lies next to the wall. There it divides mitotically, producing a large vegetative cell and a small lens-shaped generative cell, which subsequently divides and forms 2 sperm cells. The entire microgametophyte consists of the vegetative cell and the 2 sperm cells. Although extremely simple, this is a full-fledged plant. See chapters 20 and 21 for examples of gametophytes that are more complex than these of angiosperms and that are obviously complete plants. In approximately 30% of angiosperm species, formation of sperm cells occurs even while pollen is still located within the anther. In the majority of angiosperm species, the pollen is released from the anther at about the time the generative cell has formed, and sperm cells are not produced until after the pollen has been carried to a stigma. After a pollen grain lands on a stigma, it germinates by producing a pollen tube that penetrates into the loose, open tissues of the stigma. The pollen tube absorbs nutrients from the stigma and grows downward through the style toward the ovary. The microgametophyte is protected and nourished by the style tissue. As the pollen tube grows downward, it carries the sperm cells to the ovule. Almost all of the pollen cytoplasm is located at the tip of the pollen tube. The rest of the tube and the pollen grain are filled with a giant vacuole. Pollen grains are too small to store much starch, fat, or protein. If they could not absorb nutrients from the style, the pollen tubes could not grow long enough to reach the ovules. Figure 9, 11 pollen grains of Cabeye, with the pollen wall forming hexagonal ridges. The numerous holes are germination pores where a pollen tube can emerge after the pollen lands on a stigma. Pollen of Lycium has a single long groove from which the pollen tube emerges. Pollen of Macrolobium has 3 germination grooves. Figure 9, 12 in Lily, the pistil consists of 3 carpels fused together. This ovary has been cut across to reveal the 3 ovary locules or chambers. In each chamber, 2 ovules are visible. Ovules are attached to the placenta by their funiculus, which contains both xylem and phloem. The central part of the ovule is the nucellus with just 1 megasporocyte, unlike anthers, which have numerous microsporocytes. Magnification of a single ovule. Surrounding the nucellus are the integuments. Growth of the ovule has been by mitosis, but now one cell, the megasporocyte, megaspore mother cell, is undergoing meiosis. Figure 9, 13. This is the tip of a growing pollen tube. It was treated to make the nuclei fluoresce so that they can be found easily despite being so small. The lowest nucleus is probably the tube nucleus, the upper 2 are probably sperm cell nuclei, each located within a sperm cell. The 2 sperm cells themselves are located within the protoplasm of the pollen tube. Megagametophyte. Within the ovule, the surviving megaspore develops into a megagametophyte. In one type of development, the nucleus undergoes 3 mitotic divisions, producing 2, 4, and then 8 haploid nuclei all in a single, undivided cell. The technical term for a multinucleate megagametophyte is embryo sac. The nuclei migrate through the cytoplasm, pulled by microtubules, until 3 nuclei lie at each end and 2 in the center. Walls then form around the nuclei, and the large, 8-nucleate megaspore becomes a megagametophyte with 7 cells, one of which is binucleate. The 7 cells are one large central cell with 2 polar nuclei, 3 small antipodal cells, and an egg apparatus consisting of 2 synergids and an egg. Like the microgametophyte, the megagametophyte is a distinct plant. As with the pollen, the megagametophyte obtains all of its nourishment from the parent sporophyte. Plants and people. Box 9-1 Flowers, fruits, seeds, and civilization. Flowers and the fruits and seeds that result from them have always been important to us. One of the first hominids, animals on our line of evolution, was Australopithecus africanus. Fossils of its teeth, 3 million years old, show that it was adapted to eating plants. Homo habilis lived only 1.8 million years ago and likewise had a diet strongly dependent on plants. Our species, Homo sapiens, appeared evolutionarily about 500,000 years ago and survived by hunting game and collecting fruits, seeds, and edible roots. Approximately 11,000 years ago, a momentous change occurred. Small groups of humans began cultivating plants rather than just gathering them. This happened in the Middle East, an area that is now Iran, Iraq, and Syria, also known as the Fertile Crescent. The plants were common, wild species such as wheat, barley, peas, and lentils. Farming required major changes in human society. People had to stay in one place to tend and protect crops rather than follow herds of game animals. Permanent villages were necessary, as were the rules and regulations needed when people lived together in close proximity. Land, huts, and harvests are tempting targets for thieves, and thus, defense, both individual and collective, became a necessity. Government had to be created. Civilization advanced rapidly. Neolithic agricultural societies were widespread by 600 BCE, and the ox-drawn plow was in use as early as 4500 BCE. New plants were cultivated, olives, date palms, and grapes for eating and for wine. Independently, the peoples of Southeast Asia domesticated rice and soybeans. In the New World, the Incas, Mayans, and Aztecs cultivated potatoes, corn, tomatoes, beans, squashes, cocoa, pineapples, and peanuts. Table B9, 1 Locations where plants were domesticated. Figure B9, 1 Images like this occur frequently in ancient Sumerian art. The person is either a priest or a king, and the object he faces is a very strange plant. In earlier depictions, the plant is recognizable as a date palm, but through the years this important food tree became a symbol for life itself and artists began to draw it more stylized and with mystical symbols surrounding it. The person is holding a cone-like object in one hand and a pail in the other. Date palms grow as carpellate plants and staminate plants. To ensure a good crop, pollen can be collected in a pail and then applied to the carpellate plants with a brush to increase the rate of pollination and fruit production. The scene, carved before 2000 BCE, indicates that the very earliest farming societies understood this sophisticated aspect of botany. The importance of fruits and seeds to the survival of both individual humans and societies is reflected in the prominent position they were given in early art. Ancient Egyptians carved likenesses of date palms, barley, and wheat as long ago as 3000 BCE, and on Crete in 1800 BCE, Minoan artists depicted, in addition to the plants already mentioned, figs, saffron crocus, pomegranate, and lupin. They also showed several plants that must have been grown purely as ornamentals: lilies, myrtle, narcissus, and roses. As societies become wealthier, time and resources become available for leisure and pleasurable pursuits. Gardens dedicated to beautiful, fragrant flowers, not food crops or medicinal plants, arose early. Expeditions in search of exotic ornamental plants are described in the oldest epic poem in the world, the story of Gilgamesh of Sumer, and an ancient Egyptian monument, the Palermo Stone, records a plant-gathering expedition by King Snefru in 2900 BCE. Inventories list the extensive plant-collecting trips and gardens of Ramses III. Growing beautiful flowers and enjoying exotic fruits have never lost their popularity, after the Spanish discovered America, the initial exploitation was for gold and silver, but there soon followed expeditions dedicated solely to gathering plants, especially ornamental ones. Two of the many famous plant explorers sent to the United States are David Douglas, who explored the Pacific Northwest for the Horticultural Society of London in the 1820s, and the father and son team, André and François-André Michaux, sent by the government of France in 1785. Even today, gardening and growing flowers for pleasure are considered by many an essential part of our civilization. As technology increases, cultivating flowers, or at least having a potted plant on a windowsill, seems to maintain contact with our past. Figure 9:14. Shortly after landing on a stigma of the correct species, a pollen grain may germinate by pushing out part of the wall as a pollen tube. This penetrates the loose tissues of the stigma and style. This was photographed with ultraviolet light, which causes the pollen wall and collos to fluoresce and shine. Tissues of the style allow the pollen tubes to grow through them easily. Some styles are hollow and lined with a rich, nutritious transmitting tissue, other styles are solid but also have transmitting tissue. Sunflower style. These pollen tubes have penetrated deep into the style. The bright spots are collos plugs, which seal off the protoplasm at the tip from the empty parts of the pollen tube. Figure 9:15. As a megaspore develops into a megagametophyte, 3 sets of mitosis occur without any cytokinesis, thus, the megagametophyte is temporarily an 8-nucleate coenocyte. Then nuclei migrate, cytoplasm accumulates around each, and walls are established. Much of the egg is not covered by wall. The central cell is binucleate and is mostly vacuole. Table 9:3. Gametophyte development. Fertilization. Syngamy of sperm and egg involves both plasmogamy, fusion of the protoplasts of the gametes, and karyogamy, fusion of the nuclei. As a pollen tube grows downward through the style toward the ovule, it is guided to the ovule's micropyle by some means. It penetrates the nucellus and reaches the egg apparatus, then enters one synergid. The pollen tube tip bursts and releases both sperm cells, one of which migrates through the synergid protoplasm toward the egg. As it does so, the sperm cell's plasma membrane breaks down, and it loses most of its protoplasm. The sperm nucleus enters the egg, then is drawn to and fuses with the egg nucleus, establishing a diploid zygote nucleus. Because the sperm sheds its protoplasm as it passes through the synergid, it contributes only its nucleus with the set of nuclear genes during karyogamy. The sperm does not carry mitochondria or plastids into the egg, and thus, organellar genes from the pollen parent are rarely inherited by the zygote. Instead, the mitochondrial and plastid genes of the embryo are inherited only from the ovule parent. Conifers are different, their sperm cells retain their plastids while destroying their mitochondria. The zygote inherits mitochondria and a nucleus from the egg, a nucleus and plastids from the sperm cell. In angiosperms only, the second sperm nucleus released from the pollen tube migrates from the synergid into the central cell. It undergoes karyogamy with both polar nuclei, establishing a large endosperm nucleus that is triploid, containing 3 full sets of genes. Because both sperm nuclei undergo fusions, one with the egg nucleus and the other with the polar nuclei, the process is called double fertilization. The endosperm nucleus is extraordinarily active and begins to divide very rapidly by mitosis, with cell cycles lasting only a few hours. The endosperm nucleus initiates a dynamic cytoplasm and the central cell enlarges enormously, usually without cell division, into a huge cell with hundreds or thousands of nuclei. Finally, nuclear division stops, and dense cytoplasm gathers around the nuclei. Walls are constructed, thus forming cells. An example of this is a coconut full of milk. The hollow center of the coconut is one single cell, and the milk is its protoplasm. The white coconut meat is the region where nuclei form cells. A green, immature coconut is full of milk but has almost no meat. As it ripens, the cenocytic milk is converted to a thick layer of cellular meat. All of this tissue, both cenocytic and cellular, is called endosperm, and it nourishes the development of the zygote. No other megagametophyte forms as much endosperm as a coconut does. More typical examples are grains such as wheat, rice, oats, and corn, in which most of the grain is endosperm. Endosperm of these also pass through a milk stage, but by the time of harvest, the endosperm has become cellular, starch-filled, and dry enough to be hard. Figure 9:16. In many species, endosperm development is accompanied by some cytoplasmic division, and the endosperm is therefore a mass of multinucleate cells, each with variable amounts of protoplasm and nuclei and with irregular shapes. This is the only plant tissue in which nuclear and cytoplasmic divisions are so poorly coordinated, possibly because the endosperm is just a temporary tissue that is consumed by the embryo before or soon after seed germination. Embryo and seed development. As the endosperm nucleus proliferates, the zygote also begins to grow, but always by both nuclear and cellular divisions. A coenocytic stage never occurs in the embryo. The zygote grows into a small cluster of cells, part of which later becomes the embryo proper, and the other part becomes a short stalk-like structure, the suspensor, which pushes the embryo deep into the endosperm. The suspensor is usually delicate and ephemeral in angiosperms. It is crushed by the later growth of the embryo and is not easily detectable in a mature seed. Cells at one end of the suspensor continue to divide mitotically, developing into an embryo. The cells at first are arranged as a small sphere, the globular stage. The end of the embryo farther from the suspensor initiates two primordia that grow into two cotyledons in basal angiosperms and eudicots, such as beans and peanuts. While young, the cotyledon primordia give the embryo a heart shape. This is the heart stage. In monocots such as corn, only one cotyledon primordium grows out. Dicot is an abbreviation of dicotyledon, those plants whose embryos have two cotyledons. Monocots are monocotyledons, plants with only a single cotyledon on their embryos. Conifers like pine have numerous cotyledons. Later, in the torpedo stage, the embryo is an elongate cylinder. A short axis is established, consisting of radical, epicotyl, and hypocotyl. Finally, vascular tissue differentiates within the embryo. The epicotyl may bear a few small leaves, and the radical often contains several primordia for lateral roots in its pericycle. Once mature, the embryo becomes quiescent and partially dehydrates, and the funiculus may break, leaving a small scar, the hilum. In green peas, the two halves of each pea are the two cotyledons, and the stalk attaching each pea to the pod is the funiculus. In most basal angiosperms and eudicots, cotyledons store nutrients used during and after germination. During embryo development, the cotyledons become thick and filled with starch, oil, or protein, whereas the endosperm, which is supplying the nutrients, shrinks. When the seed is mature, the cotyledons are large, and the endosperm may be completely used up. We are eating mostly cotyledons when we eat beans, peas, peanuts, almonds, pecans, and other seeds that easily separate into two halves. Figure 9:17. And in many species, endosperm development is accompanied by some cytoplasmic division, and the endosperm is therefore a mass of multinucleate cells, each with variable amounts of protoplasm and nuclei and with irregular shapes. This is the only plant tissue in which nuclear and cytoplasmic divisions are so poorly coordinated, possibly because the endosperm is just a temporary tissue that is consumed by the embryo before or soon after seed germination. In monocots, the one cotyledon generally does not become thick and full. Instead, the endosperm remains and is present in the mature seed. When eating cereals such as wheat, rice, oats, and corn, we are eating almost purely endosperm. During germination, the cotyledon acts as digestive absorptive tissue, transferring endosperm nutrients to the embryo. Some eudicots are intermediate, cotyledons store some starch and protein, but a considerable amount of endosperm remains in the seed, and both methods of nutrition are used during germination. A mature seed in which endosperm is rather abundant is an albuminous seed. If endosperm is sparse or absent at maturity, the seed is exalbuminous. Figure 9:18 Embryo development. Globular stage, heart stage, torpedo stage. Embryo root, cotyledons, and hypocotyl are present. At this stage, the first xylem and phloem may become distinguishable in the hypocotyl. Mature embryo. A shoot apical meristem is shown. In some species, even some leaf primordia and a small stem are present. Figure 9:19 This bean seed has begun germinating, the radicle has extended and will develop into a taproot. The two cotyledons were digesting their carbohydrates and proteins and transporting nutrients to the root and shoot apical meristems. Beans have a well-developed epicotyl with several partially expanded leaves. One is visible here. These cells of bean cotyledon are full of starch and reserve protein. The small leaves present on the epicotyl are expanding during germination. Because they were rather completely formed before the embryo became dormant, the leaves can now mature rapidly, and photosynthesis begins almost as soon as germination is complete. The hypocotyl is curved. This allows the leaves to be dragged gently up through the soil, protected by the cotyledons. Most of a corn seed is the endosperm. The embryo is less than half the volume of the seed. During germination, the one cotyledon secretes digestive enzymes into the endosperm and absorbs the resulting monomers. Corn seeds also produce many small leaves before becoming dormant, and they can begin photosynthesis immediately after germination. The leaves are protected from the soil by a tubular sheath called a coleoptile. The amount of embryo growth and development that occurs before dormancy sets in is extremely variable. Orchids, bromeliads, and a few other species have small, dust-like seeds in which the embryo is only a small ball of cells with no cotyledons, radicle, or vascular tissue. In seeds of most angiosperm species, all of the parts are present, and the epicotyl contains 2 or 3 young leaves in addition to cotyledons. These leaves can begin photosynthesis immediately after germination. In corn, the embryo is even more advanced. It contains as many as 6 young leaves while in the seed. A fully mature corn plant often has only 10 or 12 leaves. Over half of the leaf production of the new sporophyte occurs while it is embedded in the parental gametophyte, which is itself embedded in the previous sporophyte. The embryo and endosperm develop from the zygote and megagametophyte central cell, respectively, both located in the nucellus of the ovule. Soon after fertilization, or even before, synergids and antipodals break down in most species. The nucellus expands somewhat but later is crushed by the expansion of the embryo and endosperm and is usually not detectable in mature seeds. The integuments that surround the nucellus expand and mature into the seed coat as the rest of the ovule grows. In their last stages of maturation, they may become quite sclerenchymatous and tough. It is seed coat color that lets us distinguish between black, red, and pinto beans. Figure 9:20 The seed coat of bean has an outer layer of scleriads and inner parenchymatous layers. The cotyledon is part of the embryo, and you might expect to see endosperm or nucellus between the cotyledon and seed coat. However, those have been crushed during the later stages of seed development. Fruit development. As the ovule develops into a seed, the ovary matures into a fruit. Development varies with the nature of the carpels as well as the nature of the mature fruit. The stigma and style usually wither away, as do sepals, petals, and stamens, although they may persist at least temporarily. Often 3 layers become distinct during growth. The exocarp is the outer layer, the skin or peel, the middle layer is the mesocarp, or flesh, and the innermost layer, endocarp, may be tough like the stones or pit of a cherry or it may be thin. The relative thickness and fleshiness of these layers vary with fruit type, and often one or two layers are absent. The entire fruit wall, whether composed of 1, 2, or all 3 layers, is the pericarp. Figure 9:21 in apples, the petals die and fall off after pollination. Stamens and sepals die and dry out, but they remain present, whereas carpels and receptacle develop into a fruit. In most species, sepals, petals, and stamens all upsize after pollination. Figure 9:22 A pecan nut is only the seed and innermost part of a fruit. The exocarp and mesocarp are tough, fibrous, and completely inedible. The fruit opens while on the tree, allowing the endocarp and seed to be shed. Coconuts and stores are usually just the endocarp and seed. In nature, these are surrounded by a thick, fibrous, and buoyant mesocarp and a thin epicarp. A dark spot on the endocarp is a soft spot where the seedling root and shoot will emerge during germination. Before moving on to the next section, think about a peach tree in full bloom. The tree has thousands of flowers, each with stamens and carpels. When it has finished producing pollen and megaspores, it has technically finished its own reproduction and now has tens of thousands of offspring in the form of microgametophytes and megagametophytes. Bees carry pollen from other peach trees to the stigmas of this one, and before long, it has thousands of pollen tubes growing inside its styles. You see one diploid peach tree, but inside it are thousands of haploid pollen tubes and megagametophytes. All of the water, minerals, and photosynthetes needed to keep all of these thousands of plants alive are being supplied by the leaves, roots, xylem, and phloem of the diploid peach tree. We cannot say that the haploid plants are parasitizing the tree because they are essential to its production of seeds. After fertilizations have occurred, hundreds of zygotes develop into embryos, each of which is the offspring of microgametophytes and megagametophytes and the grand offspring of the peach tree itself. This tree has other grand offspring as well, produced by its pollen that was carried to other peach trees. This tree is the ovule parent of its own seeds and the pollen parent of thousands of seeds in many other trees. Flower structure and cross-pollination. The production and development of spores, gametes, zygotes, and seeds are complex, elaborate processes, but they are not the only functions of flowers. Flowers are also involved in the effective dispersal of pollen and seeds. Because numerous mechanisms carry out these processes, numerous types of flowers and fruits exist. Cross-pollination. Cross-pollination is the pollination of a carpel by pollen from a different individual. Self-pollination is pollination of a carpel by pollen from the same flower or another flower on the same plant. In any plant population, there is genetic diversity. Random mutations will have produced some new versions of genes that offer improved fitness and some that are deleterious. For example, people with blue eyes have the same genes for eye color as do people with brown eyes, it is just that they have different versions, alleles, of the eye color genes. With cross-pollination, sperm cells and egg cells from different plants unite, resulting in new combinations of genes, at least a few of which may be better adapted than either parent. But self-pollination has about the same result as asexual reproduction because all genes come from the same parent. No possibility exists of bringing in new alleles that might provide more fitness than those inherited from the parent. However, if a plant is isolated by distance or lack of pollinators from potential cross-pollination partners, self-pollination allows it to set seed and propagate its genes rather than lose them when the plant dies. Many mechanisms have evolved that decrease the probability of self-pollination and increase the chances of cross-pollination with its accompanying genetic diversity. Stamen and style maturation times. Self-fertilization in flowers that have both stamens and carpels is prevented if anthers and stigmas mature at different times. In many species, anthers release pollen while stigma tissues are immature and unreceptive, the style may not have elongated yet, and the stigmas may be near the base of the flower while the anthers are at the top, elevated by elongated filaments. Exposed pollen lives only briefly, being susceptible to desiccation in dry air and to damage to its DNA by ultraviolet light. When the stigma and style become mature, there may be no living pollen left in the flower, thus, all pollination is affected by younger flowers just opening their anthers. This is not a very effective means of ensuring cross-pollination. On plants with many flowers that do not open simultaneously, older flowers could be self-pollinated by freshly opened flowers of the same plant. Even if pollen does come from other plants, many of the near neighbors are probably closely related because many of the seeds of a plant fall and germinate near the plant, producing a cluster of plants that are at least as closely related as first cousins. Stigma and pollen incompatibility. In many species, especially many important crop species, self-pollination is inhibited by compatibility barriers, chemical reactions between pollen and carpels that prevent pollen growth. In one incompatibility system, as a pollen tube grows, the stigma and style test proteins on the tube surface. If one of these proteins is produced by a gene that matches an incompatibility gene in the carpel-bearing plant, the stigma and style block any further growth of the pollen tube. In self-pollination, all pollen tube genes match those of the stigma and style, and blocking occurs, the plant cannot pollinate itself. Figure 9:23. The stamens of this cactus flower are mature and shedding pollen, but the stigmas are pressed together and are unreceptive. Later, the stamens wither and the stigmas spread open, ready to receive pollen from a different flower. In another common system, the critical proteins are deposited on the outer surface of the developing pollen grain by the anthers tapetum. Any match of proteins produced by the incompatibility genes blocks germination of the pollen grain. The interaction between a pollen tube and a style involves a haploid genome and a diploid one, whereas the second system involves two diploid genomes. The diploid-diploid system involves twice as many genes and therefore has a much greater probability of pollen rejection. With the diploid-diploid system, not only is self-pollination prevented, but inbreeding between close relatives is blocked as well. Monoecious and dioecious species. Among incomplete flowers there is a significant difference between flowers that lack sepals or petals and those that lack stamens or carpels. The latter two organs are essential organs because they produce the critically important spores. If either organ is absent, sexual reproduction is dramatically affected. Flowers that lack either or both essential organs are not only incomplete but also imperfect flowers. If a flower has both, it is a perfect flower even though it may lack either sepals or petals or both. Sepals and petals do not produce spores and are considered non-essential organs. Figure 9:24. This pollen grain has started to grow on an incompatible stigma, but the stigma has blocked it, causing collos to form. The collos fluoresces in ultraviolet light, so it shines brightly in this micrograph. It is necessary to consider the entire plant and the entire species as well as individual flowers. Stamens produce pollen that results in sperm production, and carpels are involved in egg production. Thus, a species must have both types of organs. Plants that have perfect flowers satisfy this requirement. But if some flowers of the species are imperfect, having no stamens, for instance, then other flowers must have stamens by being either perfect or by being imperfect because of a lack of carpels. A large number of combinations is possible. A species may have individuals that produce only stamenate flowers and others that produce only carpelet flowers. This is dioicy, and the species is said to be dioecious. Examples of dioecious species are marijuana, dates, willows, and papaya. In dioecious species, the life cycle actually consists of 4 types of plants: microgametophytes, megagametophytes, staminate sporophytes, and carpelet sporophytes. Monoicy is the condition of having staminate flowers located on the same plant as carpelet flowers. Monoecious species include cattails and corn ears are clusters of fertilized carpelet flowers, and tassels bear numerous staminate flowers. In some members of the cucumber family, the type of flower produced varies. Young plants and those growing in a poor environment produce staminate flowers, whereas older plants and those growing in a good environment produce perfect or carpellate flowers. If fertilization occurs, the carpels develop into large fruits, and only a healthy, robust plant can afford to do this. A young or poorly growing plant cannot supply enough carbohydrate and protein for fruit development, but it can supply enough to produce pollen. Dioece is an extreme adaptation that ensures cross-pollination. A plant that produces only one type of spore cannot pollinate itself. It is similar to the condition of separate genders in mammals. No individual can fertilize itself. Both conditions ensure that fertilization is by sex cells that are not identical genetically, thus increasing the genetic diversity of the offspring. Animal-pollinated flowers. The evolution of animal-mediated pollination had a dramatic impact on the evolution of flowering plants. In wind-pollinated conifers, things such as brightly colored petals, fragrances, and nectar are a waste of material and energy. But once insects began visiting early angiosperms, mutations that resulted in pigments, fragrances, or sugar-rich secretions became adaptive. For pollen that is carried by wind, the probability that any particular grain will actually land on a stigma is very low, whereas if it is carried by an insect that flies from flower to flower, the probability of a pollen grain reaching a stigma is much improved. Figure 9, 25 ears of corn are really large groups of carpellate flowers surrounded by protective leaf-like bracts. The corn silks are long styles. Corn tassels are inflorescences of staminate flowers. Each corn plant produces both ears and tassels. The species is monoecious. When this insect-flower association began around 120 million years ago, neither insects nor flowers were particularly sophisticated. Insects probably visited all kinds of flowers, not recognizing the different species. As a result, pollen often landed on the stigma of the wrong species, where it was useless. Mutations in plants that increased a plant's distinctiveness, its recognizability by an insect, flower color, size, shape, fragrances, and so on, became selectively advantageous. Rewards for the insect such as sugary nectar or protein-rich pollen and stamens were advantageous. Mutations in insects were adaptive if they increased the capacity of insects to recognize flowers that offered abundant nectar or pollen. Because it is expensive energetically for an insect to fly, the ability to recognize the most nutritious flowers from a distance while flying is advantageous. As a result, many lines of insects and flowers underwent coevolution, a flower becoming adapted for visitation by a particular insect and the insect for efficient exploitation of the flower. Coevolution has also occurred between flowers and birds and between flowers and bats. The shape of the flower is particularly important, as a pollinator actually makes contact with it. Most flowers are radially symmetrical, that is, any longitudinal cut through the middle produces two halves that are mirror images of each other. These flowers are actinomorphic or regular. But all insects, birds, and bats are bilaterally symmetrical. Only one longitudinal plane produces two halves that are mirror images. In many species, flowers and pollinators have co-evolved in such a way that the flowers are now also bilaterally symmetrical, zygomorphic. When a pollinator approaches a zygomorphic flower, only one orientation is comfortable for it. Any misalignment prevents the pollinator's head or body from fitting the flower's distinctive shape. As a result, as the pollinator feeds at the flower, pollen is placed on a predictable part of its body. When it visits the next flower, pollen is rubbed directly onto the stigma. Not only is pollen carried to the appropriate flower, but it is carried directly to the stigmas. In contrast, a pollinator can approach an actinomorphic flower from any direction, and pollen may be brushed onto any part of the body. When it visits the next flower, the pollen-carrying part may miss the stigmas and cross-pollination will not occur. Figure 9, 26. Any median longitudinal section of this poppy results in two halves that are mirror images of each other. It is radially symmetrical, that is, actinomorphic. These pentstemon flowers are bilaterally symmetrical, zygomorphic, only if cut from top to bottom are the two halves mirror images. A pollinator, also bilaterally symmetrical, fits well only if it approaches the flower properly. Anthers are at the top of each flower's throat and will put pollen only onto a bee's back. Most plant-animal relationships are a battle. Animals try to eat plants, lay eggs in them, or do other harmful things. Plants defend themselves with poisons, spines, and scleroid barriers. Mutations that permit plants to make more effective deterrents benefit the plant but not its animal pests. But both plants and animals tend to benefit from the pollination relationship, and coevolution is possible. Mutations that make a plant more recognizable or more convenient to its pollinators help both the plant and the animal, just as certain mutations in pollinators are beneficial to both organisms. But neither side should give more than it has to. Some bees now are nectar robbers, being able to get nectar from flowers without having to carry any pollen. And a number of orchids, for example Chiloglottis, trick wasps into carrying pollen for free. These orchid flowers look, smell, and feel just like a female wasp, and male wasps try to mate with them. The wasps then fly off to the next orchid flower and the next, transferring pollen between each of them. The orchids get pollination without having to provide nectar, and the wasp gets nothing more than a good time. Wind-pollinated flowers. In species that are wind-pollinated, a totally distinct set of modifications is adaptive. Attracting pollinators is unnecessary, thus, mutations that prevent the formation of petals are selectively advantageous, and the energy saved can be used elsewhere in the plant. Sepals are also often reduced or absent, and the ovaries need no special protection, thus, the entire flower may be tiny. Zygomorphy provides no selective advantage. After pollen is released to the wind, the chance of any particular pollen grain landing on a compatible stigma is small, so huge numbers of grains must be produced. Large, feathery stigmas are adaptive by increasing the area that can catch pollen grains. In general, wind-pollinated individuals produce up to several thousand small flowers. Although each flower is tiny, the entire plant has a large total stigmatic surface area. Pollination is aided by the growth pattern of the plant population. Wind-pollinated species, like grasses, oaks, hickories, and all conifers, grow as dense populations in rangelands or forests. Within 1 kilometer may be found thousands of plants and, more importantly, millions of stigmas. Species that occur as widely scattered, rare individuals must rely on animal pollination. Ovary position. The ovary and ovules must be well protected from pollinators. Paradoxically, a flower must bring a hungry animal to within millimeters of protein-rich ovules in order to effect pollination. Adaptations that maximize the separation are long styles and stamen filaments. Burying the ovaries deep within the flower provides further protection. In some species, after all flower organ primordia are initiated at the receptacle apex, the primordia crowd together, and the bases of the stamens, petals, and sepals fuse, creating a thick layer of protective tissues around the ovaries, which appear to be located below the other organs. Two terms describe this. We can either say that the ovary is an inferior ovary or that the other parts are epigynous. Inferior ovaries also can result if receptacle tissue grows upward around the ovary. The more common arrangement, in which no fusion to the ovary occurs and the ovary is obviously above the other flower parts, is a superior ovary or hypogynous parts. Intermediate, partially buried ovaries are half inferior with perigynous flower parts. Figure 9:27. Many wind-pollinated trees have such tiny flowers people do not realize they are flowering plants at all. This is an entire set of flowers of poplar, each flower consisting of just numerous stamens. Inflorescences and pollination. The positioning of flowers on an individual plant is important. Few species have plants that produce only a single flower. Instead, many flowers are produced either within a single year or over a period of many years. A large mountain ash or cherry tree produces thousands of flowers every year for well over 100 years. Many important factors determine whether a flower is seen and visited by pollinators, for example, the positions of flowers relative to other flowers, leaves, and trunk, height from the ground, and distance from an open, uncluttered flight path for pollinators. Figure 9.28 In flowers with superior ovaries, carpels sit above the other organs, as would be expected because they are initiated last by the flower apical meristem. This is an inferior ovary resulting from the fusion of the bases of sepals, petals, and stamens to the base of the carpel. Four appendages are involved whose vascular bundles are still present and distinct. In some species, after all of the appendage primordia are initiated, the receptacle tissue grows upward and surrounds the ovaries. This can be detected because the vascular bundles around the ovary are receptacle bundles, not appendage bundles. Reproductive success is measured in terms of the number of healthy, viable seedlings that become established. One important factor for this is pollination. It might seem optimal to have large flowers that can be easily seen by pollinators. Unfortunately, they can also be seen by herbivores, and because ovules and pollen are rich in protein, they are good food sources for pests. Production of the pedicel, receptacle, sepals, and petals can be thought of as packaging and advertising cost. The larger the flower is, the larger the cost. These costs can be made more acceptable by increasing the number of ovules per flower. Large flowers tend to have numerous ovules and small flowers fewer. Small flowers exist because small plants cannot afford large flowers and their accompanying large seeds, which are very expensive. Also, a large flower with many ovules is a big risk. If it is found and eaten, a major reproductive investment has been lost. However, damage to a small flower with only a few ovules is less significant. Also, if many flowers are grouped together, an inflorescence, they give a collective visual signal to pollinators. One small flower may be overlooked, but not 100 close together. Furthermore, in an inflorescence, the plant is able to control accurately the timing of the initiation, maturation, and opening of the flowers. Consequently, the plant can be in bloom and available to pollinators for several weeks even though each flower lasts only a day or two. Figure 9.29 Flowers of sausage tree are pollinated by bats, which do not like to fly among the clutter of leaves because their sonar does not work well there. Long stalks allow flowers to hang in open air where bats have free access to them. Angiosperms that grow underwater in streams and lakes usually have flowers that project above the water surface and are accessible to flying pollinators. Figure 9.30 Palm flowers are small and inconspicuous, but when grouped together, their inflorescence has even more impact than a single large flower. Thousands of tiny palm flowers are present here, each too small to be seen at this magnification. The inflorescence of combretum is even more striking than the individual flowers. Notice how the flowers mature and open sequentially. Large flowers can produce more nectar than small flowers, but this makes them a more tempting target for nectar robbers, animals that take nectar without carrying pollen. Furthermore, a flower should not produce enough nectar to completely satisfy even a legitimate pollinator because no incentive would remain for the pollinators to go to another flower. It is better if the pollinators get only enough to make them interested in searching for more. Many other factors affect the relative selective advantage or disadvantage of flower number and size as well as the number of ovules per flower. Considering the numerous types of plants, pollinators, and environments, the diversity of flower and inflorescence types is not surprising. In the simplest arrangement, flowers occur individually in leaf axils or as a transformation of the shoot apex. When grouped into inflorescences, two basic arrangements occur: determinate inflorescences and indeterminate inflorescences. A determinate inflorescence has only a limited potential for growth because the inflorescence apex is converted to a flower, ending its possibilities for continued growth. Typically, but not always, the terminal flower opens first, and then lower ones open successively. In the simplest type, below the terminal flower is a bract with an axillary flower, which also may have a bract and axillary flower, and so on. In an indeterminate inflorescence, the lowest or outermost flowers open first, and even while these flowers are open, new flowers are still being initiated at the apex. A raceme has a major inflorescence axis, and the flowers are borne on pedicels that are all approximately the same length. Catkins are similar to racemes, differing in that the flowers are imperfect, either staminate or carpellate, and all flowers of a single inflorescence are the same, and thus, each species must have both staminate catkins and carpellate ones. Catkins almost always contain very small, wind-pollinated flowers. A spike is similar to a raceme except that the flowers are sessile, lacking a pedicel. A spodix is a spike-like inflorescence with imperfect flowers, but both types occur in the same inflorescence, most often with staminate flowers located in the upper portion of the inflorescence and carpellate flowers in the lower portion, although they can intermingle. The main inflorescence axis is thick and fleshy with minute flowers embedded in it. The entire inflorescence is subtended or enclosed by a petal-like bract called a spatha. A panicle is a branched raceme with several flowers per branch. Figure 9, 31 Inflorescence types: simple determinate inflorescence, compound determinate inflorescence, raceme, spike, umbel, head. Larger circles represent flowers that open earlier than those depicted as smaller circles. Several types of indeterminate inflorescences have no dominant main axis. In umbels, the inflorescence stalk ends in a small rounded portion from which arise numerous flowers. Their pedicels are long and arranged so that all flowers sit at the same height, forming a flat disc. A head is similar to an umbel except that the flowers are sessile and attached to a broad expansion of the inflorescence stalk. Numerous bracts may surround the inflorescence during development. Heads are almost synonymous with the aster family, sunflowers and dandelions being easily recognizable examples. In this group, the inflorescences are so compact and highly organized that they mimic single flowers. What appear to be the petals are really entire flowers, ray flowers, in which the petals are very large and fused together. The center of the inflorescence is composed of a different type of flower, disc flowers, in which the corollas are short and inconspicuous. Fruit types and seed dispersal. Fruits are adaptations that result in the protection and distribution of seeds. Many different agents disperse fruits, and the seeds they contain, gravity, wind, water, and animals are the most common. The principles involved in fruit function are somewhat opposed to each other. Fruits that are tough and full of fibers or sclerids, such as pecans, walnuts, Brazil nuts, and coconuts, offer maximum protection but are heavy and expensive metabolically. Also, the protected seed must be able to break out to make germination possible. A more fragile fruit is better for that. If animals are to disperse the seeds, part of the fruit must be edible or otherwise attractive, whereas the seed and embryo must be protected from consumption. A division of labor often occurs, some parts being protective, others attractive, and still others allowing germination. Table 9, 4 agents of dispersal. True fruits and accessory fruits. The term pericarp refers to the tissues of the fruit regardless of their origin. In most cases, this is the ovary wall, but in many species, especially those with inferior ovaries, the receptacle tissues or sepal, petal, and stamen tissues may also become involved in the fruit. The terms pericarp and fruit have been applied to both types of fruit. Thus, now the term true fruit is used to refer to fruits containing only ovarian tissue, and accessory fruit is used if any non-ovarian tissue is present. Apples develop from inferior ovaries, and the bulk of the fruit is enlarged bases of sepals and petals. Only the innermost part is true fruit derived from carpels. Figure 9:32. The red, edible flesh of strawberry is really the receptacle, not carpel tissue. It is therefore an accessory fruit. The seeds are true fruits, each derived from one carpel of the flower. Pineapples develop from the coalescence of all the many true fruits of one inflorescence, so they are a multiple fruit. In addition, many non-carpellary tissues become involved, and are therefore accessory fruits as well. Bracts are visible here. Fusion of carpels also affects the nature of fruits. If the fruit develops from a single ovary or the fused ovaries of one flower, it is a simple fruit, the most common kind. If the separate carpels of one gynoecium fuse during development, an aggregate fruit results, such as raspberries. If during development all of the individual fruits of an inflorescence fuse into one fruit, it is a multiple fruit, as in figs, mulberries, and pineapple. These are also largely accessory fruits because in addition to the ovary tissue, the inflorescence axis, bracts, and various flower parts contribute to the mature fruit. Classification of fruit types. There are several ways of grouping or classifying fruits. In one method, emphasis is placed on whether the fruit is dry or fleshy. A dry fruit is one that is not typically eaten by the natural seed distributing animals. Fleshy fruits are those that are eaten during the natural seed distribution process. A further classification of dry fruits emphasizes fruit opening. Dehiscent fruits break open and release the seeds, whereas indehiscent fruits do not. For the most part, fleshy fruits are indehiscent. Animals chew or digest the fruit, opening it. If uneaten, the fruit rots and liberates the seeds. Although many dry fruits are dehiscent, others must rot, or the seed must break the fruit open itself. Perhaps the simplest fruits are those of grasses. Each fruit develops from one carpel containing a single ovule. During maturation, the seed fills the fruit and fuses with the fruit wall, which remains thin. Seeds of corn and wheat are actually both the fruit and its single seed, fused into a single structure. It has little protection and no attraction for animals. The fruit seed falls and germinates close to the parent sporophyte. The fruits are indehiscent, and during germination, the embryo absorbs water, swells, and bursts the weak fruit. These fruits are caryopses. Table 9, 5 fruit types. Beans and peas form from a single carpel that contains several seeds. The fruits, known as legumes, are dry and inedible like caryopses but do not fuse to the seeds. At maturity, the two halves of the fruit twist and break open along the two specialized lines of weakness. After liberation from the legume, the seeds are protected from small animals and fungi by only the seed coat. Fruits or seeds carried by wind must be light. They often have wings or parachutes that keep them aloft as long as possible. Such fruits are dry and weigh much less than 1 gram. Fruits and seeds that are transported by water can be larger and heavier, but they must be buoyant and resist mildew and rot. An excellent example is coconuts that float from one island to another. Animals carry fruit in a variety of ways. Dry fruits with hooks or stickers catch onto animal fur or feathers. Sticky, tacky fruits glue themselves onto animals, and fleshy, sweet, colorful fruits are eaten. Edible fruits require particular specialization because the fruit must be consumed but the seeds must not be damaged. They must not be crushed by teeth or gizzard or be digested. Immature fruits deter frugivores by being hard, bitter, or sour, such as unripe apples or bananas. At maturity, the fruit is soft, sweet, flavorful, and typically strikingly different in color. The enclosed seeds often have hard seed coats or endocarps that allow them to pass through an animal unharmed. Dinosaurs may have been especially poor seed dispersers. They had no hair for hooks to tangle in, and their digestive tracts were often too long, hot, and fermentative for seeds to survive. Grapes and tomatoes are examples of simple fleshy fruits. They are berries because all parts of the fruit are soft and edible. The seed coat provides some protection against being crushed during chewing, but perhaps more important are the size of the seeds and their slippery seed coat, which allows the seeds to slip out from between our molars. Pomes differ from berries in two ways. First, they develop from inferior ovaries, and thus, the inner tissue is the true fruit and the outer tissue is accessory fruit. Second, the seeds are protected because the innermost fruit tissues, the cores, are bitter and tough. The most elaborate fleshy fruits are drupes, in which the innermost tissues, the endocarp, are extremely hard and totally inedible, full of sclerenchyma. The mesocarp becomes thick, fleshy, juicy, and sweet, and the exocarp forms the peel, whose color informs the frugivore of the fruit's ripeness. The endocarp of a drupe is known as the pit and is often mistaken for the seed, but the true seed and its seed coat are located inside. Drupes provide maximum attraction to animals with minimum danger to the seed.